So let me introduce you to the programming, and there are several who are good programmers who are sitting with us, and there are several who are not. So it's like when it comes to programming, we are having several ideologies, and we are having some who don't know, and some who knows very well, some who knows Python, some who knows Perl, some who knows Linux, some who knows Windows, some who knows Mac OS. I'm sitting on a Mac OS teaching you Linux, but I'm yes, parallelly using Windows, and so it's programming is something which you what. So you'll ask, what is programming? So basically, I will teach you some Python programming, bioinformatics, and Linux introduction. But what is programming? Programming is nothing but uh, placing things in order. So basically, when you are cooking, you are actually doing some sort of programming. So basically, you know how to cut your vegetables, how to when to fry, when to add salt. So and the more you practice, you more better you are. So what is the top skill if you ask for a great programmer? So famous Python as famous Perl developer or inventor of Perl, Larry Wall, once said that for programming to get an expert, you need to program every day for next 2 years. If you are want to program in any language, so good at any language. So basically practice. So the more you cook your curry or dal or rice, you would become better at it, right? So you all it's so program for the programming, the skill, the most important skill is that you start reading and do not wait, just start it today, and start thinking how you can organize, and how the more you practice, the better you will be. And with this I will because my first interaction to programming was with Perl, and uh, primarily because we all worked on the sequencing strings or genomic data, which is imbibed in us. In like our body is made up of cells, and all cells are carrying a gene, and genes are strong small strings on a larger string, which is called genome. Yeah. So if you're if you're a programmer, you're from coming from IT background, you understand what is genome is. Genome is a very large string, and on large string, genes are small strings. And so basically, how big is the large string? The human genome is 3.4 GB long now. Yeah, so 3.4 GB long means one DVD you need to store. And so the big data has already covered many aspects of that. So that's why the big data is so my genome requires at least 3.4 GB to be stored. And if I have 100 joint family of 100 members, and I want to sequence genome of all of them to understand from where we are coming and where we will be going, yeah, uh, in future, and what the kind of diseases we'll have, we need 100 DVDs, yeah, minimum. And that's when the big data comes in, and that's when the programming is important. And I will discuss you theoretically like this, and also with some examples. Let's start. Okay, so let me. So when it comes to Linux, you think, okay, what is Linux? Linux is an open source operating system, and there are different flavors. And many people ask which of the Linux version I should start with. So there are in Linux is open source. So basically, today I am developing something, and tomorrow you can develop something else using your group of friends. So basically, Debian is one group on which Ubuntu. Debian was very popular, say from 2000 to 2005-6, and then Ubuntu take over from Debian. Means it was originated from Debian, and it became much more popular. So Ubuntu 20 is running now, and those who are from biology, there is a BioLinux for biological bioinformatics application. There is So the advantage of Ubuntu is, in contrary to Windows, is that in Windows you cannot label it and resell it because it's a proprietary of Microsoft. Here nobody is a proprietor. Here is open source, and it can be Abhishek Linux, it can be your name Linux. You have to invite certain features, and you have to improve and take over. So Debian is still very famous, but Debian is outcasted by Ubuntu. And in Ubuntu, there are several flavors, and one of the very famous one is Mint, Mint 20. What is Mint? Again, Mint is based on Ubuntu. Debian is Ubuntu is based on Debian. Mint is based on Ubuntu, and then a community of people, community of developers develop it for much more stable version and much more media savvy, easy to use. Suppose you are going from Windows to Linux, I will recommend you either use Ubuntu or Linux Mint. So if you want to plan installation, install one of these, and you can do it in a virtual box. Virtual box is something you do not you as a suppose you're using Windows, you can actually take over as a software. It's free of cost, and inside it you can install operating system. So basically your Windows machine is running inside that operating system will run. It can be any operating system. And so then there is a second school of thought, and there is a second school of developers. Red Hat is a company now, and Red Hat developed initially Red Hat, and from Red Hat several open source flavor has originators like CentOS and Fedora, and they are primarily Linux. And but there are some minor changes the community has made it for itself, like the way Ubuntu install software. Red Hat will not install. Red Hat will install in their own way. The commands will be slightly different. For the not overall for 90% will be same, but then 10% they will have their own improvement. It's like a famous example I will tell you. Uh, Pongal. Pongal is the uh, dish made in Tamil Nadu, but actually Pongal is not nothing but a form of a khichdi. Yeah, a khichdi, which you in north a lot of people know, and west also. So Pongal is some sort of khichdi. So basically, at the time of Pongal, entire India is celebrating khichdi. Pongal, khichdi, whatever you name. So the flavor is changed as I started adding some flavor to it, and over the years, flavors do change, and that's when operating systems develop. And there are several operating systems based on Linux, and these are two major versions. And as told you that we can do a virtual box installation. So what this is VirtualBox, on which you can go to the virtualbox.org. It's open source software now. It is hosted by Oracle, I guess, and initially it was just called VirtualBox. Now it's called open source VirtualBox, and it's a free of cost software. So actually you just download your Linux and download the software and install it in like any program, and follow the instruction on their website. You will get it. So virtualbox.org is the website. And then I'm coming to what is bioinformatics. So as the name suggests, it's informatics for resolving biological problems. So it combines biology, computer science. In computer science, it can combine mathematics, statistics. You develop software, you develop algorithm, and also those who analyze the data. Also, they are called data analysts. In this case, genomic data analyst or biological data analyst. And then uh, so combining biologists and computer scientists together, and to degree of uh, to a degree of combination. Maybe you are strong at biology and you start doing Linux programming, and after 5 years you are getting very used to it, and then you are not using Windows. So and the less you use the Windows, and if you use the Windows, you know how to use Windows for computing. Yeah, not for just writing text based solution like Excel or uh, Word. Obviously you need it, but that's not computing. That's a very basic computing, right? So uh, and then you can say, sir, I don't have my machine Linux. How can I use learn commands. So that Windows has already imbibed uh, Linux cell in it. So if you are using Windows cell 10, you can use it. There is Sigwin, which we can use for as well. And there are a lot of Google Colab will have all Python online you can use. There are several ways you can use. You can look how you can use on internet. You'll find it. So what why you need programming actually? So basically, I am taking one of the examples I show all the time, the gene prediction. So on a larger string of chromosome genome, there is a gene which you want to predict, and there is a pattern of it, right? So most of you who are coming from biology, probably you are knowing. Even if you're not knowing, then I will tell you how the gene structure look like. Gene start with ATG signal. Yeah, ATG string has to be had first ATG, then the red portion is the coding region of the gene, and that ATG the signal that now gene is going to start, and the gene will end at a signal stop codon TGA, suppose, and TGA. So that's when it will gene will end. So a linear string you're passing, and you find ATG. Okay, you can start guessing. After ATG, at a certain interval, first exon is ending, then you get a signal GT. Again, after certain length, you will get AG, so that intron will end. Then AGGT until you get a TGA signal, and full gene is there in the larger string. So uh, that's how a gene look like. So if you have to predict entire human genes, which are about 23,000 in number encodings genes, so basically you have to look this pattern. And this pattern, how you look by eyes, by hand? No, you need a kind of a calculator or a tool which can make use of it. So that's when we are using programming. So you can say a program, look for this features. It should start with ATG, it should go to GT, AG, GT, AG, and end with TGA or other stop codon. There are two more stop codon. So for example, this one. So this will pick. So it's like you have to tell your pseudocode will be start looking into the string. You have to provide your string in a text file or in the program itself, and then say search for ATG. And then if ATG is found, search next, find GT at the distance you find AG. So intron in human is much larger than fungi and all. So you can define what will be the distance between GT and AG. So you primarily on a rough scale you'll know, and that's when you will program it to know that how much distance you have to give. And by trial and error you will start finding it, or you can say any length if you get it, and then get the second pattern, third pattern, and the stop codon TGA. Then this is complete gene. And you will make some errors when you're doing programmings, and then you optimize it and you'll get the solution for it. So it's like, again, I always correlate with cooking. So or any other event is a programming. So which you continuously do and improvise it, and try to debug it. Yeah. So what in our work we have developed several automated pipelines. Okay, in one of this pipeline I'm showing, we collected fungi from marine sources. So we actually did actual sagar manthan. Yeah, sagar manthan means digging inside the sea, and we got the marine samples from the sea, and the marine fungi in this particular case, extracted marine fungi from the somewhere in the coastal sea somewhere. We send the ships for exploration, and she collected a sample, and then they bring it to the lab. Their DNA is extracted, then whole genome sequencing will happen. And on whole genome sequencing, we'll give it for sequencing by Illumina nowadays very commonly. Earlier 454, and also alternative is a PacBio or nanopore these days, and then you do de novo assembly, mapping, hybridization, and hybridization, and then you detect the repeats, and the entire process will go. So you need to program it when it comes to data comes to the computer at the stage 3 to stage 11 is analyzed on the computer. So basically you are doing the bioinformatics or computational analysis for all these steps, all these steps. So basically you need to systematically organize these workflows, programmatically or semi programmatically, so that you can repeat the analysis. The more you automate, the more you save the time. So therefore whether you program in Python or shell scripting or Perl, it doesn't matter, but or even in Java or C, it doesn't matter. But what matters is that you have a systematic way. Whenever the sample is given to you, you analyze it. And similarly, in the second analysis, what we have done is that we take the sample of insect from Europe, and then we did the similar analysis by collecting or by very similar analysis. They look differently, but this is a transcriptome sequencing. But then we have to make sure that our pipeline is working. So on a day zero, there will be no pipeline, or maybe an existing pipeline of somebody else, or from literature or from GitHub or any source. We'll take it and then we'll optimize it to our requirement, because nobody has developed based on our requirement. So we have to gauge it for our requirement and set up. So you have to. So each step is kind of a module. You can get in, get out. Yeah. So if you have a Python in Python or Perl, it's called function. Or in Python, if you have ever read a code, it's called definition, and def is a function, and then function, you need to develop a module or a function for each step, and that input of the first step, uh, output of the first step will go to second step, third step, fourth and fifth, and that's how you will start getting the result. This is the after we develop the successfully the pipeline, it looks like this. But during the pipeline, it was never linear. We were always trying different angles and optimize it, do a discussion over it, that okay, guys, in our team we have done this. There are the 5 possibilities we can do. What is the optimal solution? What is the best solution for us? And those best solution we are showing you now. But it's never. And advantage of automated pipelines are they're never static. They're evolving over time. Yeah. So today, if these pipelines were developed today, we are improving on it. And in my group, there are people who are developing improvement to this pipeline. So, and then similarly, there are several other pipelines. I mean, I'm not going into details for comparative genomics here, but you can read on our web pages, and also you will get in touch with me if you are interested. This was published in Immunobiology in 2014. And so basically, either you do programmatically or semi-programmatically or non-programmatically, but you have to develop a strategy and a pipeline. And in life, you always have to develop a strategy, right? So if Corona hit, how you will survive? You have to develop a strategy, and pipeline is nothing but your strategy. How is the stronger your strategy, the better your analysis, the quicker your analysis results. And so there are in several analyses are done, and this is our phylogenetic analysis. We are analyzing genes and putting in the evolutionary history. And then you have right side is signal signatures. And then we are doing sequence analysis and NGS analysis. So what I am showing you as on top is the sequence protein sequence analysis, and the bottom NGS sequence analysis. So there is massive application of programming in different areas of research and gene expression. So here we have taken, this is published recently in BMC, and here we are taking 4 different condition of flowering, and one condition we have developed a mutant by inserting uh, inserting tDNA tDNA, or suppose you make it some changes. So it's like I just inserted something in the naturally occurring system and then want to see the changes. So the POCO one is the changed after I shoot a gun to uh, a system, suppose, and then what you see that upper POCO one is the muted changed one, and then you see all the bars on the are on the first side. So where it is inserted, before that they have a very high intensity. In the wild or natural condition, the intensity was in the fourth axon on the right side, and that uh, that just inserting is like it's called genetic engineering. You're engineering your system, and that will show how what is the difference between flowering conditions. What is, to find a mechanism, we have done this gene expression analysis, and there also you need to program. You can use existing tools, and plus you need to program. You have to think your strategies, and then you will ask from where the data are coming. Actually, data are coming from public resources, from different data, and you are probably aware. Dr. Pankaj has already told you. And if not, you will go and look. A lot of data are available, and some data you generate locally and then analyze it. So what this line is shows that one who uh, who will not program will look faster in the beginning, but over the period, those who know program, in this red bar they are saying geek to them, they will feel annoyed because they are developing something, the process. So one very good example I can tell you that, yeah, yes, suppose you want to supply the city water, you can take it by tankers, but how many years you send it by tankers? If you develop a pipeline of water supply, actually you can sustain it much better. Same applies to programming. So if you make your water supply or your supply regular by systematic way, you will reach to an optimal level where it's normal for you. So it's when you're saying a developed country, what they have done is that's different. They have just developed a small city and replicated it to many cities. So smart concept of smart cities is also the same, that you make a if you really implement it, you have replicable cities. And so replicable programming is therefore important, that you learn how to automatize and not just supplying water by tankers, because that will not solve the problem. The day tanker will fail, your system is failed. So better to supply water with water supply with pipelines. Yeah, so same applies to the programming. Better to not today, but maybe 2 years later, you do everything by programming. Maybe your tax calculation, maybe your EMI calculation. You don't have to think, okay, I will go to Excel. Just think, can't I write it a small one in Python? So that's what is important, to have you a view about writing scripts. So again, coming back to the Linux, and Linux was originally developed by this guy. And as I told you, and this is this duck is actually duck, like organism is penguin actually. So is symbol of Linux, and this is was developed in 1991 and by a Finnish guy called Linus Torvalds. And then it becomes so popular that in the if you are in the bioinformatics, you will hear every time, are you using Windows? No, use Linux. So, and many systems use Linux, because in Linux there is no question of uh, uh, buying something. It's open source. You can build up on a lot of resources are there, and therefore people are using. And it's safe also, because the for Windows market is very big, and therefore the viruses and all will happen, and a lot of people will hear fake calls and all that they will repair their Windows, and these are there. So in Linux, the most important thing is the terminal. And so I have used my terminal, open a black box kind of things. This is and there I have just opened a text editor called using gedit. And then I have also tried to run Python here. but when I say word Python, my terminal do not understand it. And then what I did was then my script is saying try Python 3 probably, giving kind of a hint. And then I run Python 3, and then I see that Python 3 is available in my computer, not Python. And so uh, this is terminal. And what is the message here is do not fear out of this black box. Actually, you enjoy the black box. The more you enjoy it, it's your friend, it's not your enemy. And several things you can do in a command line, and it was shortly called as CLI, yes, command line interface. And so we'll see today some examples of command line interface. So suppose you are in a terminal and you want to see what is inside the terminal. So what I did was I went to my own computer. It looks very fancy, right? So basically what I did was since I'm doing writing scripts and analyzing data, so I have colored my according to my choice. And in there are ways in the terminal you can color it. So it says my computer, my bio bin. Inside I'm in a software called Hulk. Hulk is a software, and I want to see what is inside the Hulk folder, which I have taken it from GitHub. So the master option is also coming. And so it says make file read.md and run hulk.py, the Python script, src third party products. For folders are there. So if I list, so ls is a list as given above. And then if you just write ls in your terminal, if you are having terminal, you can try now also, and you can try later also. But you can get it. And if you say ls minus lhsa, just now I run when Dr. Pankaj was giving talk. So you see time, other side you are seeing the time frame, because I just learned that I have to teach now. And I like that some of the every time I present a slide, some of these things has to be totally time bound and totally new. So this time I was made, other side is saying time and the command number, so that I have an idea which one and what when I ran. And then when I am saying okay, ls star is the wild card. Yeah, so wild card like Abhishek, Kumar, and then you want to see how many Abhisheks are there in the file. You can say Abhishek star, then it will show Abhishek Mishra. Suppose I had 3,000 participant, and yesterday it was showing that some of them not put email. Last night I was sending them a reminder to join today, and I see that out of 3,000 plus, some of them put their names in email rather than putting email in the email field. And some of them forgot to put .com or Gmail. And then I have to clean those names. So basically I can use a wild type to see how many Abhisheks are there in the file. Out of 3,000, I'll just say Abhishek star, and it will print me all. So in this case I've said list any file with name star.md, list any file with name md, ending with .md. And then there was a one, as you saw in above, as you're seeing in above. So that readme.md is printed. So that's how it will be showing you the result. And yes, as I told you, this is wildcard. And obviously you can do less is, so less the file name, that will uh, show you the initial content. Less and more, you can also do more. Yeah. And so another thing is that if you do not know about a program, Linux program, suppose in this case ls I'm showing you here, you can use a manual page command called man ls. And I'm doing it, I did it just now for another terminal. So I have several terminals in my computer. So ls is equal to list directory content. So it will list directory content we saw last slide. And uh, it will also give you flags, which is the minus sign, minus ls minus if you do, what will happen. So basically if you want to learn, you will use ls minus one by one all of these in your terminal. And then you will know what is the best use and what you want to do, then according to that you will use the command. Okay, so you should give ls if you have a terminal with you, or if you do not have terminal, get your terminal and then use this command. So any command if you want to do, use. If you don't know how to use it, just do man in the Linux machine, or even in Mac it works. And then I want to change my directory. So after lsman, I just check my directory. If you want to check your directory, it will be command pwd. So it shows user Abhishek. Yeah. So user Abhishek, because I am in the Mac, and those who know Mac will know that in Mac it starts with user. In the Linux it starts with home username. And it shows that user Abhishek. And then I want to change the directory. I will say cd change directory. Shorter form is cd to document. And then I again check what is the path. The path, since I moved to directory cd, the directory documents using cd, it is now showing path of the directory to user slash Abhishek slash documents. So, and now in my terminal, there it is, the arrow is also showing that I'm in the document. So I've said you can actually go to your you can go to your bash. If you are using bash, you can go to bash.bash profile, and so there is a bash profile where you can define what you need from terminal when it is running, and or when the bash is running, or uh, any terminal is running, and there each feature will be invite from there every time computer is every time terminal is running. So there I have defined I want to see it in this red color and this font and this way. So that's the way it's showing. And this is, by the way, not mine. I forgot, I took it from internet, so I like it. So you can, it is available in GitHub. This fonting and this system is available. Maybe I can also put it in my GitHub. And again I'm coming back and I'm saying you that terminal is not your enemy, because that's when I want you to understand that you have to practice more terminal and get used to, without fearing that your computer will break down. So in the computer, as I said, the directories are there. In Linux directories, if you remember, it says slash user slash home slash user slash Abhishek, right? So in Linux, directories are placed in a very systematic order. The slash is called root or admin. In other way, root is the main organization, root. And from root has all this application folder, means there is the software will been other command line programs. Etc library, system information, users, and in my case user was Abhishek, then temp folder, and the other volumes cd e f g, what you see in Windows. And then in the Lucy or users folder, Abhishek's folder, there will be by default some directories will be there, like desktop, music, as you see in Windows. And then you can define what you want to do. Maybe you are working on a project, maybe you are working on several projects. You will first create a project folder. Inside the project folder you will have a project directory. And if you are working in a by and friends, there are a lot of guidelines how to use your project. So you in a project folder, in a general sense, also what you need in a project folder, you need a reporting directory where you can report, a script directory where you will use the scripts, or maybe to-do directory where you'll collect your to-do. So you can define your to-do list, and that will define your that will define your folders, and you can place it. By default it's called document. You can call it my document, you can tell it X document, Y document, Z document folder, or several people modify their Windows also, right? That directory system. So by default, this is the one which is obviously thrash. You need to default throw your unwanted files, and then in the script you will have a shell script that is sh text is script python script dot py, and then perl script with then test dot pl, and if the c, this is called dot c. Yeah, so or doesn't matter which programming language you have developed. And so that's what I showed you already, and that you need a command line command, then option parameter and the file name, and then it will bring you the options, and that's what it has done in the pwd, as we have already seen. And then if you want to change directory, which we have already did earlier, uh, that we have so the I will answer the questions later. So there's some question coming up, but I will quickly go because maybe I'm losing the time. Yeah, so I guess that is for signal for time. So yeah, so there are change directory. You can change to /home /anil. You will reach the anil folder, and if you say dot, that means you are in it will show you are in the current, you want to make changes in the current directory. If you do double dot, one directory back, that means if you are in an anil, if you say cd dot dot, you will reach one directory back, means you will reach home if you are in an anil. Okay, so make directory is the, you want to create the directory, make directory test. Then you want to move something from one to other, one file to another file, or one folder to another folder. You say move source through destination, and you can do destination. So in this case, I'm saying move file 1 to file 2. So file 1 will be not be there anymore afterwards, and it will become file 2. And then second case, you want to move file to directory. You are making, so that will be from wherever it is, it will go to the directory, inside the directory, and move one file from one place to another file within the directory. And copy is the, again you copy file 1 to file 2, source to destination. And yeah, source copy file from one directory to the specified directory. And you can do recursive copying, and then you can also man cp, yeah, and test it when you are trying. And so these are some examples. I'm not avoid time. So if you are removing file, you have to be very careful, because removing will delete your files which may be in you requiring it. So in Windows also you have to be very careful when you are removing files. And removing directory will be rm minus r, and rmdirl will also remove directory, but the funny thing is that only remove empty directory. So therefore it's of some use but not fully of use. So better to use rm minus r. So I'm not going to the content. And if you want to concatenate, you concatenate by cat command. And like concatenate means suppose you have file 1, file 2, you have a 50 email id in first file and 15 second file. You want to have a fun email id, all 100 email id into same file. You say cat file 1, file 2, and you can create file 3 or print on the terminal. Okay, so I will skip this, and then obviously you can have a user read write command. So if you understand Linux, you will start understanding that if Abhishek has permission. So it's similar in Windows 10 also nowadays, that you can only, user can only use some portion of the operating system if they have permission to use it, read write and execute. So we'll skip this permission now, and then because you have to do some programming test as well. The time is going. Gedit of text editor is it is. And the programming, why Python or programming is required, I already told you. Python, let's me come to the Python. Okay, and if you write a program in assembly language, this big it will be. When you write in C, it will be this big. And you write in Python, it will be one line. So result equal 10 plus 5. So like if you want to compute sum of 5 and 10, in Python it's one liner, and assembly is multi, maybe 8 or 10 lines. In C it's at C 5 lines. So basically the more lines, the more chance of error, and more difficult to learn and remember. And so therefore Python is simpler. That's what the whole idea was. And Python was developed by Gudovan uh, Rosem, and obviously history you can read. But what I want to show you that there are two versions of Python. One which has come to an end this year, to Python 2, and Python 3. If you are starting from as of today, you can use Python 3. And obviously you can use several text editor, gedit, text wingler and if you're using Windows, you can use notepad plus plus, and in terminal you can use Mac or Linux, you can use terminal for writing program there. But there are several ways for using Python nowadays. In a 45 minutes time I cannot cover all, but we do run a course over 2 months or so, so that those we can enjoy if you are interested in joining our team on getting full training. So the Windows in Windows you can use PowerShell, or also in Windows you can use now Linux terminal is available in Windows 10. So and obviously on a terminal you write Python 3. That's what I did right here in the right side. And then yeah, so therefore I when I ran the Python script, I ran Python and then Python 3, and so Python 3.8.2 was running in my by default was available in my Linux machine. And when I want to run my script, I can say python 3 dot python.py. So here we are showing you one example. If I am running print hello world, print hello world, and say in the program actually I have on the screen I have written it, modified it, and said just print hello. It says print hello, but when I say /n twice, it printed by two new line character saying hello, and that is new line. Since it is a new line character, it is also printing. If you want to print tab, you just use /t hello world, or /t hello. In this case I have printed on the screen. You see that hello is printed. So the way you want to print, you can print. The way you want to modify your data, you modify your data. So you can yeah, so you most of the time you see shashbang user bin in Python 3 or something like this. That is for telling a machine which Python from where to take it. And also for user to know which Python I'm using. Yeah, so that in your computer, if you're a complex user, you may have several Pythons, and therefore you want to know which script is using which Python. And so here what I have done is that for those who are biologists, I have taken some messages coming. Just a minute. Uh, okay, so some private messages coming. I will answer those questions later. Let me go to the script. Oh, sorry, I'm the chat now. Okay. So yeah, so what I have done here, I have taken a large protein script. Okay. And then we want, I said this sequence, a small sequence first. Protein equal to, in the bracket, I put this sequence. Yeah. So you can just put your name, Abhishek or Jat, whatever is equal to protein. And then you can, in Python, you want to know the length. You just say length, the string where you have put the, in this case, protein. I just said length, get the length in the bracket protein, and it will give me length of 23 amino acid. Okay, and then I just say dir. It shows me the what all functions I can inbuilt function of Python I can use with this. So there is a lower, and I said lower, and when I said lower, something happened. But then I said lower dot py, then something happened, nothing happened. Actually, I made an error. But then there is a way in which you have to do it. So my syntax was wrong. I was not pulling if you pull syntax wrong, I'll make an error. So then what I did was I just take out the put it like protein dot lower, and then in the lower I see that protein, small protein equal to capital protein dot lower, and bracket means all the input will be taken out. So you see that the 23 characters, which is larger in caps in upper portion, became a smaller uh, small letters. Yeah, it's lower letters. It converted to lower letters, and you can take any protein, put it like this, as I have put it, and practice this, some of this dir variable name, and then you can try some of the this, and lot of this will work actually. And so this was a live session where we had practiced it, and from there I have taken the example. This was done on the uh, 1 month earlier. Maybe I have some more example for that. But what is important here, that you start using Python on a terminal, and uh, and the Python full course on the Python will be available on YouTube as well from Mr. Manish. And then if you want to use it, you go to the Python in an interactive cell, and you can use help and get help from the suppose you don't know what print is doing, say help inverted commas print, and then print will print, and you can go to online doc tutorial and say 3.2 tutorial, and then it will uh, place your queries. And with this I think I've taken a lot of your time. I hope that I have put your stimulated you to learn programming. My whole idea is to give you an insight, and this is what the first for beginning when somebody is joining my lab, I will provide them this kind of ideas, to discuss on ideas and ask them to do it, and we check whether they are if they are not knowing. If they are knowing, they have different, then they have different requirement, then they have to directly come in and develop databases for us. But those who don't know, they are also very useful in other sense. They will if I have asked you for interview, I know where I can use you, otherwise I will not even waste a minute. Yeah. So there you will know that. And then I'm now answering your questions. Let me stop the video.